start studying the cell biology of the membrane trafficking started long ago. I don't tell you when, but long ago enough. And so in these years I've been studying different segments of the membrane trafficking compartments. So starting with the regulated segregatory system, but then soon after moving to the Golgi complex, that has been the topic of my studies around this year. And more recently I'm also studying some aspects of the endocytic system. So needless to say that the endomembrane system is a very important one for many important and crucial functions in the cell. So the importance of this membrane trafficking system has been highlighted, of course, by the recently awarded Nobel Prize to these three scientists for their discoveries of machinery regulating vesicle trafficking. That is a major transport system in our cells. So, you know, the work by Jim Trotman, Randy Sheckman, and Thomas Sudoff, and essentially they won the Nobel Prize for three key aspects of the field. So Rotman was the first to isolate COP1 vesicles from the Golgi membranes, and to discover the mechanism that is responsible for fusion of vesicles with the acceptor compartment, that are the snares proteins. Sheckman isolated a number of yeast mutants that had problem in generating or in consuming vesicles. So he had this collection of hundreds of mutants, and he understood that the problem in consuming vesicles or forming vesicles were due to the lack of genes that were important player in the mechanism of vesicle formation and fusion. And Thomas Sudoff studied how this basic machinery that these people had identified has an important role in neurons. Okay, so the molecular machinery that have been identified by these three people and their group are really key to the process of membrane trafficking. But there is a limit. The limit is that these molecular machinery have been identified mainly in reconstituted systems. For instance, Rotman, in his seminal studies, had these membranes coming out from cells that were homogeneous, and it was reconstructing in vitro the system of intra-Golgi trafficking. And if you use this in vitro condition, what you see are usually small vesicles, and indeed this is what Jim Rotman states in his lecture when he was receiving the Nobel Prize. At the end of his talk, he gave this kind of an argument to the many dozens of contributors to our understanding of the tiny bubbles, small vesicles, that in the cell enable all thought and action. Okay, so the main trafficking intermediates, if you go in vitro with homogeneous membranes, are represented mainly by these small vesicles. But in the reality, this is not the case. I mean, small vesicles are only a very tiny fraction of the possible transport carriers that mediate trafficking in the cell. So if you after these seminal discoveries, applying the different technology that allowed to follow trafficking in intact cells, it has been clear rather soon that indeed the carrier were not just small vesicles, but they were much more complex. And this term was introduced of pleomorphic carriers, meaning that sometimes the pieces of membrane that are responsible for trafficking are indeed very complex membrane that include tubular portion, cisternar portion, and of course this also opened the question on the mechanisms that are responsible for the formation of these large carriers as compared to the ones that are responsible for the formation of small vesicles. and we will see something related to this later on. But of course, you have big carriers such as phagosome or micropinosome, or of course secondary granules, in which you have a big piece of membrane that contains hormones, for instance, or enzymes. Not only this, so this is a kind of trafficking that is called dissociative trafficking, in which you have a piece of membrane that detaches from a donor membrane and that eventually fuse with an acceptor membrane. So you have a kind of dissociation of a piece of membrane from a donor and then a fusion with the acceptor compartment. But you might have many, this is just to tell you that, I mean, this is a scheme, a drawing, but if you go and look at the arm of the transport carrier, for instance, this is what you can see as for the COP2 dependent carriers, that are the ones that originated from the endoplasmic reticulum. So this is what you see in vitro system. You see these nice, regular, small vesicles. But if you go in cells, for instance, and you follow the trafficking of a cargo out of the endoplasmic reticulum, a large cargo, such as, for instance, procollagen, and we'll go back to this later, you see that the kind of structure in which the collagen is incorporated is not at all small vesicles. The procollagen fibers, when they leave the arm, they are much larger than as vesicles. They are usually 300 nanometers, whereas a vesicles is 60 nanometers. So the reasoning is very simple. So a cargo of 300 nanometers cannot be accommodated in a vesicle of 60 nanometers. And in fact, what you see, and this is seen by making a 3D reconstruction of EM tomography, and you can see here this chunk of red. That is a carrier containing collagen that is very different from the small vesicles that you see in vitro. So just this to give you the concept that when you look in the cells how the different cargo are trafficked, you realize that the small vesicles are only one of the possible mediators of the trafficking. But I was also coming to the point that not only you have this dissociative way of trafficking in which you have budding of a carrier from a donor compartment and fusion with the acceptor compartment, you might have also different strategies of bringing a cargo from one compartment to another. So this is just the vesicular dissociative way of trafficking, but then you have also another way to move a cargo that is by compartment progression or maturation. This is a process that has been studied at least in two compartments. One is the endosomal system and the other one is the Golgi complex. In the endosome, you know, when a cargo is uptaken by the outside, by possible different possible main way of endocytic entry, then it reaches the early endosomes. And once in the early endosome, the cargo progresses through the late endosome, and this kind of progression does not occur through the budding of a vesicle from the early to the late endosome, but the cargo remains in the early endosome, but it's the early endosome that changes its identity because it loses some components that are peculiar of the early endosomal system, such as, for instance, a kind of ramp, the ramp 5, and acquire different components, such as, for instance, ramp 7, that is a feature of the late endosomal compartment. So in this case, it's not the cargo that is leaving the compartment to go from one compartment to another, but it's the compartment that is changing its identity, and the cargo always remains in. And this is also what in the Golgi complex has been shown to occur. So originally the work made by Calade and then of course by Rothman in vitro was suggesting that the cargo moves from the cis Golgi cisterna to the medial via vesicles. So that eventually a vesicles forms from the cis Golgi and then fuse with the medial Golgi and then with the trans Golgi. But again, just for a very simple reasoning, such as considering some very large cargo, in this sense procollagen is a paradigm, again, procollagen cannot enter small vesicles, so it has to go through the Golgi by a different way. And so the way that many groups, but essentially the group of Alberto Luini, identified was this cisterna maturation. So the concept is very similar to the one that we have seen for the endosomal system. So the collagen is here in the cis Golgi cisterna, and never leaves the cisterna. It always is in, but it's the cis Golgi cisterna that acquire a different identity because it is receiving material from the medial cisterna. And what is making the difference between a cis Golgi cisterna and the medial Golgi cisterna are essentially the glycosylating enzymes, the glycosyltransferase or glycosidases. So what happens is that the secretory cargo remains in the cis cisterna, but the cis cisterna is receiving enzymes from the medial cisterna via a retrograde trafficking. So this is a way by which the cargo then will end up to be in a medial kind of cisterna, no longer in a cis kind of cisterna, without exiting the lumen of the cisterna. This is a concept of maturation that, these are the two clearest examples, but there are many other possible examples in the trafficking. And then there is also another possibility that you have a direct fusion of a compartment with another. So there is no vesicles in between, there is no maturation, but just that the entire compartment fuse with the acceptor compartment. This is what, for instance, the late endosomes do when they reach the lysosome. In that case, you have a late endosome that is fusing with the lysosome without any kind of intermediate. And finally, this fusion can be very transient, it can be very stable, or can be transient, just the time to transfer the content. And this is the mechanism of a kiss and run. And finally, you might have continuity existing between different compartments. This has become clear with the advance in technology that we had in the last year. So when we've been able to do 3D reconstruction of EM tomography, in which you can follow the compartment in the 3D space. You can analyze not just thin sections, in which you will never be able to pick up a connection between compartments, but you can reconstruct entire areas of the cells using the EM tomography. It has become clear that many compartments are connected by tubular continuities. And this is, for instance, has been seen also in the Golgi complex, in which some of the cisternae are connected by tubular continuity. Okay. Just to say that membrane trafficking is not just the vesicles that go from one side to another. It's more complex, and there are many ways to traffic cargo from one compartment to the other. And of course, this kind of process, trafficking, is mediated by molecular machinery that can be very complex, and they include many different components. And what it has become clear is that mutation of these components of the molecular machinery are causing disease that affect many different systems. So here what I've collected are just a list of genes that when mutated give rise to different disease according to their intracellular distribution. So these are genes encoding for protein that are component of the membrane trafficking machineries. And so you have many in the endolysosomal compartment, in the Golgi complex, and the endoplasmic reticulum. So these are all the disease genes of the membrane trafficking pathways. And just giving a closer look, you have genes that are encoded for protein that are involved in the trafficking at the level of the endoplasmic reticulum, for instance, in the R to Golgi trafficking pathway, or in the trafficking pathway of the plasma membrane. And I sent you a paper in which we have studied one of these components, that is the TRAPC2. I don't know if it will be one of the journal club that we have this afternoon, or you have chosen the other one. Okay, so this is, for instance, mutation of this component, this complex that is responsible for this spondyloepiphyseal dysplasia target, but you will tell us later on what this is about. And I mean, there are many components of the endocytic system that, when mutated, give rise to many different diseases. So you have many diseases affecting the nervous system, and you have also disease affecting the kidney. This is the other disease we are studying, and I will talk today of some of these of our studies related to the OCRL. So this is the other paper. So you are going to present also the other paper?
No, just one.
Okay. So I will tell you something about this, and also connected to this, the mutation in CLC5 that gives rise to a similar clinical outcome. Okay, so this is just to say that as a cell biologist, we can learn a lot from the study of Mendelian disorders, because the example I was giving you, just to study a simple molecular machinery in vitro reconstituted system, is of extraordinary importance to get, I mean, the hands on the basics of the mechanism. But if you don't apply the basics to the physiological context in which the machinery is operating, you really are just at the beginning of understanding the process. And this is what I think we can learn from the study of the Mendelian disorder, because you realize rather soon where in the organism that molecular machinery is really important. Just studying the signs of the disease and essentially going and study the relevant cell system. So I will tell you what we have learned studying, for instance, the role of the OCRL protein, but we could learn this because we went to the cell system that is the one that is affected in disease, that are the kidney cells. So if we had to choose the wrong system, we couldn't understand anything about it. So just trying to understand the basic mechanism of Mendelian disorder will give you the physiological context in which you have to put your molecular machinery. And on the other side, as a cell biologist, if we understand more on the mechanism of the disease, we can give a lot to the understanding of the disease, but possibly to the identification of targets for the cure of this disease. So I'm just giving you some of the examples of what we have learned as a cell biology from the study of genetic disorders. For instance, this is the eye cell disease. It's a lysosomal storage disorder called eye cells because it's an inclusion cell disease, because the cells from these patients have a large inclusion inside. It's the mucolipidosis type 2. And studying what was wrong in the cells from these patients, it was understood that there were no problem in encoding the different lysosomal enzymes. So there was no mutation in the lysosomal enzymes. Still, the lysosomes were not working. And it was found that the cause of this dysfunction was due to mutation in the machinery that is responsible for adding the mannose-6-phosphate to the lysosomal enzymes. So you know that the lysosomal enzymes, of course, are made in the endoplasmic reticulum. They go to the Golgi. But then at the trans-Golgi network, they are specifically targeted to the lysosome because they bear this tag that is a mannose-6-phosphate that is recognized by the mannose-6-phosphate receptor and that sort these enzymes in specific carriers that are delivered to the lysosome. So this is the way by which a lysosomal cargo, while it is at the trans-Golgi network, knows where to go. Otherwise, here from the trans-Golgi, it could take one of the possible way out to the plasma membrane, to the secondary granules. But since it has this tag, it is recognized by its receptor and it is brought to the right compartment. So what happens in this eye cell disease is that there is a mutation of the enzyme that are adding the mannose-6-phosphate to the enzyme. So the lysosomal enzymes are properly folded, but unfortunately they cannot reach the lysosome and are secreted because they have not the right tag. Okay. And so, studying the disease, we understood the mechanism of the targeting of lysosomal enzyme to the right compartment. Another important example is this of this protein that is AGIC53. So ERGIC stands from ER, Golgi Intermediate Compartment. So this is a protein that, as a cell biologist, we have been using for years, just as a marker of a compartment, because it is very nicely localized to this compartment. It's in between the ER and the Golgi. But then it was found that indeed the mutation in this, and so we were using it as a marker without realizing its function. It was very useful. All of us used, at least until 10 years ago, without knowing what it was there for. But then the disease was, the mutation was identified in a disease that is characterized by combined deficiency of factor V and factor VIII. And here the mutation was a mutation of one component of ERGIC53. And it was realized that indeed ERGIC53 acts as a cargo receptor for this soluble coagulation factor that is responsible for the trafficking of this coagulation factor from the ER to the Golgi complex, and then, of course, for the secretion of this coagulation factor. So when ERGIC53 is mutated, this coagulation factor cannot be exported out from the endoplasmic reticulum because they lose their receptor. Okay? Because ERGIC53 is binding the factor 8 and 5 in the lumen, and on the other side, ERGIC53 brings the signal to be exported out of the endoplasmic reticulum via the COP2 vesicle. So if there is no receptor, the coagulation factor will remain in the endoplasmic reticulum and will be eventually degraded. So again, so we understood the function of a protein in the cell by studying the disease in which this protein is mutated.
Sorry, Antonella. I guess that factor V and factor VIII are made in hepatocytes.
Yeah.
So what's ERGIC53 doing in other cells?
Yeah, this is, you should know, is Bob, what is studying Bob here. Bob Sidia is studying because, I mean, this is for sure the relevant function in the organism because when you have mutation disease, but it is also, for instance, a cargo for ERP44. But possibly there you have a redundancy of systems. So when you don't have ERGIC53, you might have something else that can help ERP44 to exit the ER. But this is the only receptor for this in the hepatocyte. Okay, so this is another example. And then I want to give you an example, but this is the one that we'll study in the paper you are presenting this afternoon. And this is connected with the role of the COP2 machinery in the ER exit. Because there, you know, you will tell us that the COP2 machinery, everything is triggered by the small GTPases SAR1 that when activated recruit the coat complex that is made by an inner layer made by sector 2324 and an outer layer made by sector 1331. And you have here a list of animal models or human disease that are caused by mutation in the single components of the COP2 machinery. And you can realize that the consequence in the organism of mutating these components that are ubiquitous are very different. For instance, mutation in this SAR1B will give rise to what is called the Anderson disease or chylomicron retention disease, whereas mutation in the SEC23A, for instance, will give rise to a craniolenticular pseudodysplasia. So here there is a problem in the osteogenesis in the skull. Here you have a problem in the secretion of chylomicrons. So this implies that each of these subunits and each of these isoforms seems to be a cargo specificity. So this is what we learn from the study of human disease, that not all of them are the same in terms of cargo specificity. So we learn that different cargo have different requirements in terms of COP2 components. And studying the different disease, we can also try to understand why these different requirements are established and how these components control the COP2 cycle. But we can skip this. The concept is that you understand the specificity of the machinery studying a given disease. And finally, this is another great lecture we are learning from the study, lesson we are learning from the study of genetic disorder, because recently studying disease in which you have the common clinical phenotype that is caused by mutation of different genes, you can reach the understanding of molecular complexes that work together. For instance, this is the case from of the hereditary spastic paraplegias, in which you have mutation of many different genes that give rise to the same clinical phenotype. This imply and suggest, and in some cases it has been demonstrated, that the product of these different genes are acting together in the same molecular pathway. And for instance, this is the way by which it was found that spastin, ratlastin, and RIP1 work together in shaping the endoplasmic reticulum. So these are three genes that, when mutated, give rise to the same disease. Originally, their function was not understood, but then it was found that they indeed exist in a complex, and they are controlling the very same basic mechanism, that is, in this case, the shaping of ER tubules. And this is also what we are trying to apply to another group of disease, group of mutation that gives rise to a common clinical phenotype, that is the renal Fanconi syndrome. So renal Fanconi syndrome is just a dysfunction of the proximal tubular cells in the kidney. So the main function of the proximal tubular cells is to reabsorb salt and low molecular weight from the filtrate. And there is a condition in which this reabsorption is not occurring properly. And this gives rise to the so-called Fanconi syndrome that have these common signs. Of course, you have impaired absorption of bicarbonate, phosphate, urate, low molecular weight proteins. And you have, of course, systemic consequences of this impaired absorption, especially at the level of the bone, so you might have rickets. And so far, many different conditions have been identified of Mendelian disorders that, in which the single genes are mutated and the clinical phenotype is very similar. So these are mutations, for instance, of the transporters that are present on the luminal plasma membrane of the proximal tubular cell. So mutation on these different transporters have been shown to give rise to the renal Fanconi syndrome. And interestingly, mutation in proteins that are in the endocytic system, such as this intracellular chloride channel CLC5 and this phosphatase OCRL, have also been shown to be responsible for renal Fanconi syndrome. And this is what we have been studying. We have been studying the mutation in OCRL that give rise to the Lowe syndrome. Lowe syndrome is a syndrome in which you have the renal Fanconi signs, so the impaired absorption in the proximal tubular cells, but you have also oculo and cerebral sign. They are born with congenital cataracts, 50% of them just develop glaucoma, and there is a central nervous system impairment with central hypotonia and secondary retardation. And so this is a very special project in the lab in which we are not just working at the bench, but we are also helping the family of the Italian Lowe syndrome patients. So when I was working at the Consortium Mario Negri South, it was essentially more than 12 years ago. I was invited to a meeting at the NIH organized by the American Association of Lowe Syndrome, because as I will tell you in a while, I'm working on phosphoinositide. So I was invited at that meeting, and at that meeting the president of that association told me, look, in Italy the situation is desperate, because from time to time we are contacted by families that do not know any other family in Italy having the problem with having this kind of the disease. So you should do something. So I came back and just contacted through telephone the mother of one of these kids, and we started the Italian Low Syndrome Association. So at the beginning there were three families, now there are 40 families. We meet every year, and we have this kind of association in which only clinicians are present. Essentially they are ophthalmologists and nephrologists. And I mean, this is very helpful and for the families, and recently the Italian Low Syndrome Association has joined the European Low Syndrome Federation. So I mean, that's just to tell you that it's a special project in the lab, in which we are also looking at helping the families. But what is OCRL? So mutation in OCRL gives rise to this low syndrome. OCRL is a phosphatase, but is a lipid phosphatase that is acting on phosphoinositides. I guess you know what phosphoinositides are. They are minor component of our members in terms of quantity, so they are phospholipids that altogether account for 10% of our phospholipids in the cell membranes. And they are called phosphoinositides because their polar head is made by inositol, and inositol can be phosphorylated in one of the possible positions 3, 4, or 5. And so you can have different phosphorylated species of phosphoinositides with one phosphate, and this might be PI3, PI4, PI5 phosphate, with two phosphate, and maybe here you know the most famous one that is PI45P2 or PIP2. Maybe you know this. Or you can have the only three phosphorylated species, that is PIP3, that is the one that is made by PI3 kinase, that is important in inflammation, in cancer, and so on and so forth. So the phosphoinositides are important not only because they can be precursor of second messengers, such as, for instance, PI45P2, that can give rise to diacylglycerol and IP3, as all of you know, but because they are important components of our membranes, because they control different machinery that are involved in membrane trafficking or, for instance, in actin cytoskeleton remodeling. So this is just a closer look to the phosphoinositides, and just to tell you what OCRL is doing. It is a 5-phosphatase, so that is removing the phosphate in position 5, and so bringing back PI45P2 to PI4P. Okay, as simple as that. But why is this important? Because the different enzymes, so there will be PI kinase and PI phosphatase. Each of them is mediating a specific step of phosphorylation or dephosphorylation, and for each of them many different isoforms in the cell. So altogether the PI kinase and phosphatase are roughly 50 proteins, a family of 50 proteins, and these proteins are differentially distributed in our cell. And so the balance between the activities of the kinase and phosphatase is different in every cell compartment. And so the final concentration of the different phosphoinositides turns out to be different in the different compartments. And this is what, a simplified scheme of the different intracellular distribution is such that, for instance, the PI45P2 is mainly present at the plasma membrane, and it is present under basal condition, whereas, for instance, the PIP3, the only tris-phosphorylated species, is present at the plasma membrane, but only after stimulation with growth factor, for instance. The lipid that is more represented in the endocytic system is the PI3P, so it's a monophosphorylated species with the phosphate in position 3. And so you will have it in the early endosome, and you might also have PI35P2. So this is the domain of the PI3 phosphoinositides, and of course autophagy is another process that is very much dependent on PI3P.
Sorry, Antonella.
Yeah.
How do you actually locate these components?
That's a very good point. So you can locate this because there are protein domains that maybe most of you have encountered so far, that are, for instance, the Plex homology domain, or the PX domain, or the NTH domain, that can rather specifically recognize the different phosphoinositides. So that's also why these phosphoinositides are so important, because they can recruit on a specific membrane a given set of cytosolic proteins. So they are giving a sort of identity to our compartment, because depending on the phosphoinositides, you will recruit a given set of proteins and not another one. So not only this, but you can use these protein domains fused, for instance, with the GFP, and you can transfect this domain in the cell, and this domain will go where its phosphoinositide is more concentrated. So this is, here it is, now how we can build this map, just to answer your question. So if you have your domain that has a good specificity for a given phosphoinositide, such as, for instance, PI45P2, you just make a chimera of the domain with the GFP, transfect this domain in the cells, and this will localize mainly on the plasma membrane. So this will tell you that that phosphoinositide is mainly at the plasma membrane. If you, for instance, transfected a pH domain that has an affinity for PI4P, it will go mainly in the Golgi, where the PI4P is more represented. But, of course, there is a limit, because, first of all, in order to see the lipid, in this way you have to transfect the cells with the domain. And you know, this is not completely innocent. So this domain by itself, if it is overexpressed, it might also cause some kind of redistribution or alteration of the lipid itself. So you have to use caution in interpreting the results using these domains. However, this is the only way we have so far to try to map the different phosphoinositides. For some of these lipids, there are also good antibodies, and so far there are good antibodies for PI45P2, PI4P, and we start having for PI3P. Of course, you have to label the cells, and you have to be very cautious in keeping the cells on ice, because this is a lipid, it's not a common antigen, so it's very labile, cannot be fixed, so you have to be very fast, and you have to do everything in cold room, so that's why it's not very nice to stain these lipids. However, so in order to map the different lipids, either you transfect this with the GFP chimera, or you can also make this domain recombinant in, for instance, E. coli. And in this case, you make them fused with GST, so you will have your GST lipid binding domain, and you can use this as an antibody, for instance, on cryosection. So you can incubate your section of the cells that you want to study with this domain, and then you will add an anti-GST antibody, and you will label the section, and you will see where the domain is localized, and presumably the lipid is localized. Okay. That's just to go to the concept that I was mentioning you, that a kind of identity code of the different cell compartment can be established based on the kind of phosphoinositides and RAB. You know, these small GTPases are also very specifically distributed in the different cell compartments, and one possibility, so this might be a kind of philosophical question, why, I mean, an early endosome is different from a late endosome, what gives the identity to our organelles? Of course, on one side is the content, because each organelle has a particular environmental content, but on the cytosolic side, a very important determinant of the identity is given by a given combination of a phosphoinositide and a given small GTPases. Okay. So what I want to tell you today, I mean, just to give you, to tell you how we are approaching the study of the role of OCRL in the cells, and trying to understand what are the molecular pathways that are more important for the function of OCRL, and to also see if we can identify the pathway and possible correctors of the defects that are induced by OCRL mutation. So we have been using these three different approaches. So a classical approach of the cell biologist, we have an hypothesis and we want to test this hypothesis. So we have been studying the role of OCRL in membrane trafficking in the relevant system, as I was telling you, so in the kidney cell. And this is the, I will go briefly through this because this is published work so far. Then I want to spend more time on what we are doing in this direction. So we are exploiting our knowledge in the cell biology of OCRL to set up a cell-based assay to be used in the screening of library of compounds, small molecules, to look for some of these small compounds that is able to correct the cell problems that are caused by a defect in OCRL. And if we have time, I want also to tell you what we have learned by applying an unbiased approach to the problem. So we are studying, in this case, what happens to the cell when OCRL is not working, without having a specific hypothesis in mind, but just to study the gene expression profile and to see how the cell reacts to the loss of OCRL. So, of course, these approaches are very complementary at the end. Just to tell you a few words on OCRL. So OCRL is an enzyme, is a phosphatase, but is a very complex molecule. So one of the groups that have more advanced our knowledge of the structure of OCRL is the group Pietro De Camilli, who has crystallized all the domains of OCRL. So in OCRL you have a phosphatase domain, and you have other domains that are an N-terminal pH domain, a low gap domain. However, what I want to stress is that there are many motifs in OCRL that are responsible for the interaction with the component of the endocytic system, such as the adaptor complex 2 or the clathrin. And so far, OCRL has been localized to different cell compartments, essentially the plasma membrane, the early endocytic system, and more recently also to the primary cilia and the Golgi network. Our studies, as I was telling you, have been concentrating on the role of OCRL in the proximal tubular cells. That is the cell that is not working in the patient. So we decided to go and to look there for the role of OCRL. And this is, the scheme of a proximal tubular cells in which, on the luminal side, you have to reabsorb a lot of stuff going to the tubules. And for sure you have to reabsorb low molecular weight proteins that are filtered by the glomerulus. And this kind of reabsorption is really a pure clathrin-coated mediated endocytic process. Because the low molecular weight in the lumen are bound at the surface of the proximal tubular cells by a receptor, that is the multi-ligand receptor megalin, that belongs to the family of the LDL receptor protein family. So megalin capture the low molecular weight proteins, and there are many of these. That's called multi-ligand because it combines many different ligands. Then megalin and its ligand are internalized in clathrin-coated vesicles, go to the endosome. There, for the acidic endosomal pH, ligand dissociates from megalin. So the ligand goes through the lysosome, is eventually degraded, and then is exported to the basal portion of the cell, whereas megalin is actively recycled to the luminal plasma membrane, apical plasma membrane. So in order to have an efficient reabsorption, you have to have a very efficient recycling of megalin from the endosome back to the plasma membrane to make many rounds of internalization. What we have found is that, so I will go just to the conclusion of the paper, What we have found is that OCRL is important for this megalin recycle. What happens when, so this is, for instance, what happens in control condition. You have, for instance, your ligand that is bound to megalin, that is internalized by traffic-encoded pits, vesicle goes to the endosome, and then go back to the plasma membrane. What happens when OCRL is not working is that megalin, you have no problem in capturing the ligand on the surface, but then you have problem for the megalin recycling from the endosome back to the plasma membrane. So you have a kind of what we call the traffic jam in the early endosome. So megalin gets to the early endosome, but then cannot exit the early endosome. Why is this, of course? And this is not just megalin, because other receptors that have the same trafficking pathways are also trapped in this early endosome, such as, for instance, transferrin receptor, or other receptors that also go through the endosome to reach a different destination, such as, for instance, the mannose-6-phosphoreceptor that has to go from the endosome back to the Golgi, is also trapped in the early endosome, or also the EGF receptor that has to exit the early endosome to go to the lysosome is also trapped there. So you have a problem in exiting the early endosome. And why is this? So what we could show is that when OCRL is not working, there is a problem in the distribution of phosphoinositide. So if you think to what happens during an endocytic process, so there are many steps. Of course, you have to bud a piece of plasma membrane inside to make a coat, to make the vesicles, and then to deliver the vesicles to the endosome. But among the many different things that have to happen, one concerns the phosphoinositide, because here you have to go from a membrane that is enriched in PI45P2, that is the plasma membrane. You have to end up in a membrane that is enriched in another phosphoinositide, that is PI3P. So you have to consume in some way the PI-P2, and you have to produce the PI3P. And what we have shown is that OCRL is very important in operating this kind of phosphoinositide switch. Because if OCRL is not there, you keep having the wrong phosphoinositides on the early endosome. So you keep having a lot of PI-P2 in early endosome. And this is because you don't have the phosphatase that should remove these phosphoinositides. And having this PI-P2 in the wrong place is a problem. And we understood that at least one of the reasons of this problem, that is you stimulate actin polymerization. You have a lot of, an excess of polymerized actin, filamentous actin on early endosome. And this, of course, impairs the exiting of cargo out of the early endosome. Okay? So we understood that OCRL is an important controller of this phosphoinositide switch in the endocytic process, and if you alter the balance of phosphoinositide in the early endosome, you alter the balance of actin polymerization, and this causes problem to the trafficking out of the endosome. But now, I mean, this is published work, we are now trying to understand more on the details, but we thought at that point that maybe we had learned enough to try to exploit this knowledge to find correctors for this condition. And so we started this long pipeline that is a pipeline that you have to go through if you want to apply an high content screening approach. So what is the high content screening? High content means that you make a screening based essentially on imaging. So usually the high throughput screening are based on biochemical assays. Now you measure enzymatic activity, receptor binding. Here what you are measuring is a cell phenotype. So it's a very rich content because you can have a different aspect in this phenotype. And then you want to use your phenotype in a very unbiased way to try to find something that corrects your phenotype. Okay, so this is the simplest step. But you have to go through all these steps. So the first step, that is the crucial one, is you have to identify a cell phenotype. And so this cell phenotype has to be very robust, because then you have to use this cell phenotype to go through all the pipeline. So it has to be very robust, because then you will screen thousands of compounds on that phenotype. So you have to be very sure about it. And so this was the first step. So we had cell phenotypes. We had endocytic phenotype in these cells in which OCRL was mutated or was silenced. And so we thought maybe we can exploit this cell phenotype to go through this pipeline. And one of these phenotype just is the consequence of this endocytic traffic jam that I was mentioning you, in which, for instance, the mannose-6-phosphate receptor is trapped in the early endosome instead of coming back to the Golgi. And this means that if you look at this, if you look at the distribution at the steady state of a mannose-6-phosphate receptor in control cells, you will have this nice perinuclear localization that is mainly Golgi complex, because at the steady state, the mannose-6-phosphate receptor is at the Golgi complex. But if you look at the distribution of mannose-6-phosphate receptor in cells depleted for OCRL, you will see that now the mannose-6-phosphate receptor is very much at the periphery. So this is a clear phenotype. So the peripheral distribution of mannose-6-phosphate receptor that here is trapped in the early endosome, as compared to the control condition in which the mannose-6-phosphate receptor has gone back to the Golgi. Okay? And this is a very robust phenotype. So peripheral distribution of mannose-6-phosphate receptor. And so the simple idea was let's screen conditions such as different small molecules or different siRNAs to see if we can find something that can bring back this phenotype to this phenotype, to correct the sick phenotype. Of course, since this is a very long way to go, you have to be sure that the phenotype is relevant for the disease. Otherwise, you are looking something that is just there but has no importance. And so one approach we took to understand if it's relevant was, first of all, to look in cells from patients. And you can have these proximal tubular cells collected from the urines of the patients, because there is a normal shedding of the cells from the tubules, even in control subject. And especially in these patients, you have this shedding is more pronounced, so you can collect the cells from the urines. And we could see that also comparing the distribution of mannose-6-phosphate receptor in control versus patient cells, we had a similar peripheral distribution. So it was something that was observed in cells from patients. And in addition, this was a kind of finding that was known since a long time, that in the serum of these patients, there is an increased concentration of lysosomal enzymes. And this is what we see also in cultured cells, in which these cells secrete lysosomal enzyme in the medium, because the mannose-6-phosphate receptor, so we talked this about this morning at the beginning of the lecture, is not there at the trans-Golgi network to bring the lysosomal enzyme to the lysosome. It is out of place, and so the lysosomal enzyme, once at the TGN, are secreted. So this is an indirect sign that the mannose-6-phosphate receptor is not operating properly. So once we had validated this assay is relevant for the disease, of course then we had to optimize the assay to have this phenotype reproduced in cell lines that are easy to handle, of course. And these were, for instance, ELA cells in which we used the inducible knockdown of OCRL. And then, of course, you need the right tools and the right microscope. So these are automated microscopes that allow to analyze hundreds of samples, looking at sample in 96 well plates or 384 well plates. So you seed your cells in this, we've done this in 384 well plates. And the microscope automatically will go from one well to another, acquiring images. And so there are different systems. So the one that we have at TGEM is this from Perkin-Elmer. There is a big one that is called Opera, and the other one is called Operetta. They are all Opera-inspired instrument. And there is another one that is from Nikon. So what you do, what you have to teach the microscope the phenotype they have to recognize. So first of all, you have to give some kind of reference point to the microscope. So in order that the microscope understand what is the object that it has to analyze. And usually you enable the cells with a nuclear staining. So the microscope will understand that each nucleus will correspond to a cell. And then you will label, for instance, with your marker. In this case was mannose-6-phosphate receptor. And so the microscope acquired the images, many different images for each well. And then the image analysis is such that the microscope will measure the structure that are labeled in a given channel. For instance, here we use the red channel for the mannose-6-phosphate receptor. And we'll give you a series of parameters that are the number of the spots that we measure and the distribution of these spots compared, for instance, to the nucleus. And so you can then, without going into much details, you can then have a kind of distribution in population of your cells according to the presence of the mannose-6-phosphate receptor spot. For instance, in 4 categories, you can have the perinuclear localization of the spots that is very much similar to that of the control cells, in which the spots are all in the Golgi, so you have this perinuclear continuous structure. Or you might have just a peripheral spot. You might have a combination of the perinuclear fragmentation with peripheral spot, or just perinuclear fragmentation. So having all these images, you can classify your cell population in the classes that are resembling what you have analyzed from your phenotype. And so, for instance, if you classify the cells in these 4 classes, you will see, and you just compare the control cells with the OCRL knockdown cells, you will see that whereas in the control condition, the majority of the cells follow in this class 1 category, that is perinuclear continuous structure, in the OCRL knockdown cells you have a dispersal of the population because you will have fragmentation and presence of peripheral structure. So you will have a different distribution of the OCRL depleted cells as compared to the control cells. And then you can then see, can we find something that is able to revert this phenotype, can bring back this mannose-6-phosphate receptor to the perinuclear localization. And okay, I will skip this. So what we have done, we have screened a library of 1,280 compounds, small molecules. These is a library of pharmacologically active compounds from Sigma. Here you have many enzyme inhibitors, receptor antagonists, ion channel blockers. So compounds that are either FDA approved drugs or just pharmacologically active. And we have screened this library on our phenotype. And this is what came out from the first screening. Here, each dot is a drug, is a compound, and you have to compare the position of each dot to the position of our OCRL knockdown cells or control cells. Here we are measuring the population of cells that have a perinuclear continuous staining. So the mannose-6-phosphate receptor is in the Golgi. So these are the control cells. These are the OCRL knockdown cells. And each of these spots corresponds to the phenotype of cells treated with these compounds. As you can see, just looking all together, the majority of these compounds are not affecting the distribution of the mannose-6-phosphate receptor. So many of them are not affecting significantly, some of them are even worsening the peripheral distribution. But there are few of them that looks very interesting because they are bringing back the sick phenotype to the, let's say, control phenotype. So they are able to rescue this peripheral distribution of the mannose-6-phosphate receptor. And of course, now that we have this, so these are roughly 100 compounds that seem to be interesting after this first screening. And then, of course, we had to test these compounds in secondary screening, in secondary assay, that are more relevant also for the disease. So we have screened these 100 positive hits on other assay, for instance, the uptake of megalin ligand, to see if they were able also to rescue this kind of endocytic impairment in the cell. And we found that out of these 130, roughly half of them were also able in rescuing the uptake of megalin ligand in proximal tubular cells. So they were really interesting. And I mean, these are the hits that are confirmed, and they belong to these different categories. And what now we have to do is to go to the last step of the pipeline, that is to test some of these hits. So we are now performing further tests, of course, on this to really choose the best ones. And once we have at least 5 or 6 compounds out of this, we will test them in animal models of Lowe syndrome that are available. And so just this is to say that starting from, understanding the cell pathway in which your gene of interest is working, then you can identify a relevant phenotype, and you can exploit this phenotype to find correctors. And this is just a list of other conditions in which some cell phenotypes have been identified, and this might be exploited for a similar kind of approach. So I don't know, I would leave out the last part of the talks just to leave enough time for the question.
Whatever you want. Whatever, you choose. You choose.
No, if there are questions, I would be more happy to. If not, I can give you.
You show a very nice picture about this